In today's video, we're looking at gel electrophoresis. This is a technique we can use to separate out molecules by either their size or their charge, and is useful for all kinds of things like DNA fingerprinting or in clinical chemistry to separate out proteins. We're using a demo today where some short pieces of DNA have been digested by a restriction enzyme. So we're assuming now that you're familiar with the theory behind how gel electrophoresis works. So we're focusing here on the lab techniques that we need to analyze the DNA. Let's do it. Okay, now before we start, there's a piece of equipment here that you might not have come across before that we need to be using quite a lot during this process. So we're going to have a look through that first and understand how to use that properly before we begin. So this is called a micropipette. This is a piece of equipment which is used to pick up and transfer very small quantities of liquid very accurately. So this is an adjustable one. So you can see on the side here, we've got a little window which has a number in it. That number is the number of microliters that this pipette is being set to pick up. And the little red number at the end is a decimal point. So this can measure to the nearest tenth of a microliter. That's the nearest 10 millionth of a liter. So a very, very small volume of liquid that it will transfer very accurately. So what we do is, first things first, we need to set this. Now the first volume of liquid that I'm going to need to transfer in this lab is 20 microliters. So I'm going to turn the top of my micropipette until my number is 20 on the side. There we go. Now, this also needs a tip on it. This is without a tip, has a little metal nib at the end. The tips come sealed in plastic. Now, it's very important that we don't touch the fine end of these tips. Any oil on there will ruin the surface tension of the liquid inside, and the measurements here are so precise that changing the surface tension of the water in there will mean you get a different volume of liquid. So we always handle these from the outside of the bag, get one of them pointing upwards, tip of the micropipette inside, push the tip on, and we're ready to go. Now, this micropipette has two stops on the button at the top. So if you push this down gently, you will come to some resistance in the first place. You can then push through that stop all the way to the second hard stop. These are sometimes referred to as the soft stop and the hard stop. The first stop, the soft stop, is the one we're most interested in. That is our precise volume based on what's on the window. All right, so if I wanted to pick up 20 microliters, I've set my micropipette to 20. I push down to my first stop. I'm then going to put the tip into my liquid, release, and I will have 20 microliters in here. The second stop is for when we're depositing the liquid, so we can push all the way through and make sure that we don't have anything left in the tip at the end. So we're all set up now. I've got my micropipette set up. We're going to pick up 20 microliters with that as it is now. So my first job is I'm going to put the DNA sample into my restriction enzyme to digest it. And because the DNA samples are all different, they'll have different recognition sites for my restriction enzyme along their length. That means that once they're digested, they'll be in different sized little fragments, and so they will run differently through the gel. So I've got my 4 samples here. I've got A, B, C, and a reference sample. So these are all labeled on my microtubes here. These are being frozen, so the first thing I need to do is just to thaw them. So just between my fingers, I'll just hold these until the DNA sample on the inside has thawed. We're then going to need to mix them. So the DNA has a tendency to stick to the plastic tube. So the first job here is going to be to pump the liquid inside the microtube just a few times, just to mix up the DNA and the water again, so that we get a nice clean sample that's well mixed. So here we go. Now, because I've got different DNA samples here, I need to make sure I'm using a different tip so that we don't cross-contaminate them. So we're going to get through a few tips, but that's absolutely fine. That's why these are disposable. To remove the tip from here, this darker grey sort of paddle on the side here will release the tip. So we're going to put it over. We've got a beaker here that says use tips. Let's put it over the top, push down this paddle, and the thing will pop off. And then we can use a new tip for our next sample of DNA. So once we've mixed our sample, we now need to introduce it to the restriction enzyme. So we're going to take 20 microliters using my Micropipette into my little tube of restriction enzyme, which I have over to the side here. So I've got 4 of these little tubes for my 4 different samples, and I'm just going to introduce them into there. And again, give it a good mix in there to make sure that this is all mixed up before we do the digestion. Now it's really important this is really well mixed. That can introduce some air bubbles if you're unlucky, so I have a few here. If that's the case, then you can just throw this into a little microcentrifuge and spin it out, and that will get rid of any of the bubbles on there. If you've just got a couple, then sometimes just giving it a tap on the desk will get rid of them, but otherwise spin it in the centrifuge and no harm done. So we're now going to label this, put it into the water bath at about 37 degrees for about 20 minutes. That'll allow the restriction enzyme to do its thing, digest the DNA, cut it up at all those restriction sites, and then we'll be able to compare our samples using the gel electrophoresis. So my 4 samples have been in the water bath now for about 20 minutes or so that digestion will have finished. What we're going to do now is to add 2 microliters of what we call a loading dye to the DNA samples. This is just going to show us whereabouts on the agar gel the DNA will have got to after a certain time, so we know when to stop the gel electrophoresis. So I'm going to reset my Micropipette to 2 from 20 as it was earlier. So my window now says 2.0. That's going to pick up 2 microliters of the loading dye, and using a separate tip for each of my samples, remember we don't want to cross-contaminate, I'm going to put 2 microliters of this loading dye into each of my blue tubes. And again, use that technique to mix it, right? That pumping just to the first stop to mix up the dye with the sample. One thing that's quite key when you're mixing is only ever to go down to the first stop. What that will mean is that when you take your pipette back out again, to get that last little bit out from the tip of the pipette, you can go down to the second stop and that'll empty it. If you mix by going all the way down through to the second stop each time, you'll have no way of emptying the very last bit from your pipette. So yeah, only ever mix going down to the first stop. Okay, so I've got all 4 of my samples now well mixed. They've got the loading dye in there, they've been digested, they're ready to go into the wells, all right? So this is the next stage of here. We have got an agar gel at the bottom of this tray. It's been set with this comb in here so that we have those 4 wells across the side of it, which are cunningly here labeled 5, 6, 7, and 8. 1, 2, 3, 4 would have done, but 5, 6, 7, and 8 is okay. So first things first, we need to take the comb out in order to leave the wells behind. So it's really important here to be removing the comb really carefully so that we don't tear the gel as we remove the comb. So we'll put it flat on the desk, and we need to be pulling the comb straight up vertically in order to leave our wells behind. So nice and gently, straight upwards, and that'll leave our combs behind. It can also help at this point to put this whole thing onto a black piece of paper that'll let you see the wells much more carefully. So I'm going to do that now. So we now need to pipette 20 microliters of each of these samples into one of the wells on the gel. So we've got 4 wells in the gel and 4 samples. Remember again, we're going to need a fresh tip for each of these because we don't want to cross-contaminate anything. So 4 samples, 4 tips, 4 wells. Let's do it. So we're going to readjust my Micropipette to 20 microliters. I got 20 from my first sample. So it's sample A. And I'm going to put that into my well. Now, to get this accurately, we want the pipette to go into the liquid that's covering the gel, but we don't want to touch the gel because it's very delicate and we don't want to tear it. So it's very easy to puncture. So use the side of the gel to rest the pipette tip so you keep everything nice and still. We just want to go just ever so slightly under the surface of the liquid, just breaking the surface tension of it so that the loading dye, which is more dense than the buffer in the tray, is going to help the DNA to sink to the bottom of the well before it starts to be drawn along it. So I'm going to use both hands here, using the side of the tray as well to stabilize everything, and just hover the tip above the first well, break the surface tension of the buffer, and then push all the way through to the second stop. to completely fill the well. And when I see an air bubble forming at the end of the tip, I'm going to lift it out. Okay, so that's one sample. I'm going to use a new tip, 3 more samples to go. Okay, so that's my wells filled. I'm now ready to fit this with my electrodes and draw that DNA through the gel in order to separate out the fragments of different size. Okay, so to do that, we've got a 36-volt power supply all wired up and ready to go. We're going to use some carbon fiber electrodes. So we're going to put an electrode at each end of the gel. And now DNA is a negatively charged molecule, so it's going to move towards the positive electrode. So once we've got our 2 electrodes in, one at either end, we're going to make sure that my negative pole from my power supply, which is usually the black wire, is going to go at the negative end, so the DNA end with the wells, and my positive electrode is going to go at the opposite end. What that will mean is that the DNA is going to run from negative to positive. So it's going to draw the DNA through the agarose gel towards this negative electrode. This is where the loading die gets useful as well, because the loading die is also drawn towards the negative electrode, so it shows us where the DNA might be and tells us when to stop running the gel. So when the loading die gets almost to the end of the gel, we're going to stop it, disconnect the power, and then we're going to develop that and see where the DNA is. Okay, so we ran these for a couple of hours with the power on to draw the DNA through the gel. Once we'd switched those off, we then stain it with a stain called toluene blue. That's this one. This is going to stain all nucleic acids, a really nice blue color, so that it will show up on the gel. The DNA itself is relatively colorless, you won't be able to see it in the gel, so you need the stain in order to show you where the DNA is. That's then left to develop overnight, and then this is what we end up with after about 12 hours. And you can see in this gel the different bands of the DNA where the toluene blue has stained the nucleic acid, which will show you then the relative lengths of those DNA fragments. So you can see you've got different fragments from the 4 different samples because they had recognition sites for that restriction enzyme, which was BAMH1, at different points along their DNA code. So that's that. That's gel electrophoresis. So that's how we separate molecules out by size, particularly with DNA, using the fact that DNA is negatively charged to draw it through the gel. Thank you for sticking with us. There's a link to a written version of the protocol in the description down below. Have fun with it, and we'll see you next time.